wherever possible, it's important to try to implement comprehensive next generation sequencing. Now, understandably, this cannot be done at many laboratories and requires volume and requires an appropriate business case. So there's always the option to send out testing to an appropriate laboratory. So I'm going to just give you our perspectives as a molecular reference laboratory and some of the reasons why we went down the route of comprehensive next generation sequencing. And as Dr. Vanderlyn mentioned, there are a number of targets. And so to be able to most cost effectively capture those alterations in those different targets, you have to start thinking about high throughput DNA, RNA sequencing and a panel that assesses for the diverse alterations in multiple, sometimes up to 100 genes at once. Why hundreds of genes? Why do we need to be looking at hundreds of genes? Well, you need a lot of genes to sequence in order to be able to report out a high quality TMB score. And so that's one of the reasons why we've now evolved to comprehensive panels. So it actually results in cost efficient utilization of limited tissue, and tissue samples can be very limited in the context of lung cancer. And the goal is to yield as much actionable information that is going to help drive therapeutic decision making, but there's also diagnostic and prognostic information that comes from next generation sequencing.